Hello and welcome to today's virtual short course where we'll be helping you empower cancer immunology research with CEDAR. My name's Nina Blazeska and I'm the Senior Project Manager of the Cancer Epitope Database and Analysis Resource. I'd like to thank you all for joining us today from whatever part of the world you may be in. I'll be giving a brief overview before we hand it over to our wonderful presenters for today's short course. But the first question you may be asking is, why are we hosting a 3-hour short course today? Well, there are multiple reasons. The first being it helps us engage with our global user community because we are finding more and more people from all around the world are using the Cancer Epitope database and its tools. As you know, today is a 3-hour short course, and some of you may be familiar with the 3-day course that we run for the Immune Epitope database. But today we'll be focusing in on the cancer resources, the database tools, and provide you with some research examples. It also gives us an opportunity to improve our resources through getting some user feedback through our interactions today. It also helps us answer your questions through the multiple Q&A sessions that we'll have over the course of today, and it will help hopefully facilitate learning about our resources. And then lastly, the short course will help us share ideas to further scientific research. We've got a wonderful cast of people today that will be presenting that you'll get to hear from. Shortly, I'll hand it over to Dr. Alessandro Sette, the Principal Investigator of the CEDAR resource, followed by Dr. Bjorn Peters, the co-PI. You'll also hear from Dr. Hannah Carter, a professor and collaborator of the Cancer Epitope database from the University of California, San Diego. You'll also hear from Dr. Zeynep Kosologlu-Yalçın, our instructor for CEDAR, Dr. Matthew Bussey, the Senior Curator of the database, and Dr. Ibel Kari, our Bioinformatics Postdoctoral Fellow. Very briefly, the short course is structured via a series of presentations from everyone I've just mentioned, and we will have multiple Q&A sessions after the presentations to help answer your burning questions. So to that end, we please ask that you add your questions to the Q&A function in the Zoom webinar. That will help our speakers pass through the questions a little easier. And then I will post a link to the slides and a couple of other resources in the chat, and then also our end of day survey. So all of that will be in the chat. And we do ask that you save your questions for the Q&A rather than the chat, as it just helps our speakers manage them a little more efficiently. And then lastly, you can follow along with subtitles by enabling the transcript. Okay, an overview of the agenda. So shortly, Alex will be presenting the introduction to CEDAR, and then Matt will be speaking about the curation and database structure. Alex and Matt will conduct the first Q&A, and then you'll hear from Bjorn and Zeynep on how to access the data, query reporting, and examples. They will host the second Q&A, and then Ibel will be presenting on the next generation tools and cancer pipelines. You'll then hear from Hannah on a tools cancer study, a case study. Ibel and Hannah will conduct the third Q&A, and then again you'll hear from Zeynep on the cancer meta-analyses, and Zeynep will wrap up the day with the last Q&A. So we should end by 12 p.m. Pacific time today. Couple of other event logistics, you will receive a post-event email, and in that email there'll be a link to the workshop recording, and the presentations will be shared with you in the chat shortly. Likewise, the survey we will be sharing with you. Your feedback is important to us because this is our first official CEDAR short course, and we'd love to keep improving for next time. And lastly, if you are using the CEDAR database or any of our tools, please be aware that there may be some slowness due to some increased traffic. But lastly, please do follow us on social media. We are on Blue Sky at CEDAR-epitopes, and then we are also on X at CEDAR_epitopes. We'd love you to share what you've learned today or something interesting about your research, and don't forget to tag us. And now, without further ado, I'd like to introduce Dr. Alessandro Sette to start the introduction to CEDAR. Thank you very much, Alex.
Thank you, Nina, for that very nice overview. And let's jump in in terms of introduction to CEDAR, I would like to start by pointing out a bit of history. Advancing the slide, Nina. Thank you. So really, this is put in context of the two sibling databases. One is the IEDB, already mentioned by Nina, and then CEDAR, which is what we are concerned with today. And the IEDB has been in existence for over 20 years, since 2003, supported by the NIAID, the National Institute of Allergy and Infectious Diseases. And consequently, the focus of the IEDB is epitopes and data from infectious disease, allergens, autoimmune diseases, and transplantation, and so forth. What was missing from the IEDB was the data related to cancer epitopes. And this was changed in 2021 when National Cancer Institute, the NCI, started to support the effort ongoing for CEDAR and allowed us to develop a sibling database specifically, uniquely focused on cancer applications. So what is CEDAR? In the next slide is shown a overview of what are the content of CEDAR, in the sense that there are two components. One is the database, and the other is the analysis resource. So the database catalogs cancer epitopes that are described and published in the literature. And very importantly, the cataloging is based on the context in which the epitopes are described, and the data, the underlying data, is actually captured in the database and accessible to the user to customize, also, as we will hear later, their query and extract the data that is relevant to the user. And the analysis resource is a compendium in which the user can operate tools. Some of them are shared with the IEDB, others are more cancer specific, to do analysis or predictions using their own data, or perhaps even the data that they extracted from the database. Next slide quickly lists the motivation for the CEDAR project. First of all, epitopes and cancer immunology is an ever-growing field. Epitopes represent the molecular structure that is recognized by adaptive immune response, and adaptive immune responses play a key role both in the process of the cancer development and regression or disease, and also in immunotherapy. And so it's important to have these tools and understanding of what is described, what is available, to allow to molecularly define what are the events associated with cancer and immunotherapy. There are, as highlighted in the bullet number 2, there are several resources that catalog cancer epitopes, but we noticed and realized two key features. One is that the existing resources do not allow the user to actually extract the context, and this lack of granularity is a real issue in some of the applications, and from our point of view, and also limited computational prediction analysis tools, of course. And in fact, we have been aware throughout the years the fact that many users of the IEDB actually used the database and the associated analysis resource specifically for cancer research, even though the IEDB was not necessarily designed to address the needs of the cancer community. The next slide emphasizes the fact that in the development of CEDAR, we have been able to jumpstart our efforts because of the IEDB 20 years of experience. And this capacity to jumpstart the project is manifested in several different aspects. One is the fact that we have had procedures and SOPs to already well defined in terms of how to curate the data. We had existing tools. We had a established user base. So we were able to really capitalize with great savings in time and effort in terms of establishing the CEDAR database and analysis resource. So next slide, just want to point out that CEDAR is a neutral resource. And what do I mean by that? Is that in the context of CEDAR, we don't have a value judgment of what is good data, what is bad data, what is a good epitope or a bad epitope. We capture the experimental data that is underlying a published report that describes a cancer epitope from XYZ. And pretty much like in PubMed, when you make a search in PubMed for, I don't know, a certain tyrosine kinase, you don't want to hear what necessarily the PubMed staff thinks are good papers or bad papers. You just want to know what's available. And so this is exactly the same philosophy that we have at CEDAR. We make all the data and all the epitopes that are described available to the user, and then the user can actually filter and query the database to customize what kind of data the user wants to extract. The next slide underline a aspect of CEDAR, which is its versatility. And this is manifest in several different aspects. One, that the CEDAR staff is very involved in community standardization efforts, so that whatever is captured by the CEDAR database is captured with nomenclature and fields and ontologies that are consistent and compatible with what is generally developed and utilized in the scientific community. And in fact, the CEDAR ontologists play a key role in further developing this common language that is used throughout different resources. At the same time, in terms of resource interoperability, it's important to underline that CEDAR takes advantage of this common language and this interoperability. So we take advantage of taxonomic and CBI and other GenBank, other resources that provide and maintain different aspects of the information that is captured in the fields of the database. And lastly, last but not least, today you will hear from Zeynep how the data that is captured in CEDAR with increasing volume of data, as we will see in a second, can actually be used to gain insights in specific areas of research. And we have today two examples, one related to prostate epitopes and the other related to neoepitopes. The next slide points out the growth of information in the June 21 to June 25 period. So as you will see, the y-axis of this graph starts at 4,500. And why is that? Because, again, underline how we were able to hit the ground running here. The first thing that we did was to be able to import all the data that was already in the IEDB that is related to cancer-associated viruses, such HPV, or in the case of hepatocellular carcinoma, hepatitis B virus, and so forth. So that was immediately populated. And then we got busy. We started to curate cancer epitope proper data. We privileged initially prostate and neoepitopes. We also make sure to immediately curate any new paper that is published, regardless of what particular cancer epitope is or category is related to that paper. And also we started to go back in time. So to curate papers from year 2020 and 19 and so forth. And so we are now current from year 2025 all the way back to 2013, and we keep on curating. And so we have an estimated, it's going to be about another 2,000 references, considering the backlog going back from 2013 to the start of time, so to speak, and also keeping current, of course, and also getting into curating epitopes from animal models that currently have not been tackled yet because we privileged the human data in our initial period of curation. So in the next slide, you see the growth of the visits to CEDAR over time. And at this point, throughout the life of the project, we have about 40,000 total site visits, and with a monthly median visit to CEDAR in about 3,000 visits. So it's a growing user community. Today is a very important day for us because we are holding this first workshop. And as Nina highlighted, we want to hear from you. We want to hear from you on what, if you use CEDAR, what you found worked well for you, what did not work well for you, because we want to learn and make it better. And also, we want to just make you aware, if you have not used CEDAR, of what are CEDAR's capabilities and opportunities for you to use it. Next slide, we also have published papers highlighting in the press the capability and the existence of CEDAR and some of its initial meta-analysis that we've been performing. And this has been received with an increasing number of citations to these publications. So we look forward to seeing the numbers in 2025, and we anticipate that it's going to be an ever-growing number of citations, and we strive to make the CEDAR visibility go through peer review reports, because we like the rigor of the process of peer review and keeps you at that, holds you to a high standard. Next slide, just pointing out the fact that ultimately one of the fundamental goals of the IEDB and CEDAR is to catalyze applications. So on one hand, we certainly want to support, as one of our primary goals, the basic research in cancer immunology. And at the same time, we also want to enable people using the databases and analysis resources to do their work. discovery, product development. And in fact, we've been very pleased to see that many different patents actually quote the IEDB or CEDAR as having been using these resources as part of their discovery or product development process. And we also support this. We have a specific program that allows companies to license the IEDB so that they can use it, or CEDAR, but so they can use it in the confidentiality behind their firewall and so forth. So in summary, in the last slide, key CEDAR functions and achievement, continued growth of data. There's about a little short 400,000, little short 500,000 epitope structures, more than 1 million, 1.3 million to be exact, assays that are linked to these different structures, and about 6,000 different studies on various cancer and related topics. As I was mentioning, the CEDAR is neutral and versatile, community ontology, presentation of the data as this is published, interoperability across different resources, a growing user community with a median of 3,000 monthly site visits, and a growing impact judged as from the reference that quote the CEDAR resource, and also the existence of patents that use this concept of a database and analysis resource. And with that, I thank you for your attention and having listened to this introduction.